Many signal transducing receptors are plasma membrane proteins that bind specific extracellular molecules such as growth factor, hormone, and neurotransmitters, and then transmit to the cellular interior a signal that elicits a specific response. These responses are usually cascade of enzymatic reactions, giving rise to many different effects within the cells, including change in gene expression. Membrane-bound receptors are classified into major three groups. Ion channel linked receptors, which is similar to the potassium channel. Second, G-protein linked receptors and enzyme linked receptors. The last two has three domains: the extracellular domain and transmembrane domain and intracellular domain. It's very difficult to determine the receptor structure. In most cases, just the extra domain and the intra-domain structure were just determined. We have no detailed structural knowledge on how signals are transmitted through the membrane, nor on how receptors are linked to ion channel signaling. Receptors that trigger many responses have two things in common. First, their transmembrane regions contain 7 helices. Second, signals transmitted to the intracellular domain of these receptors are usually G-protein. You can see some example mediated by G-protein. Epinephrine binds to the beta-adrenergic receptor. Its effector will be adenine late cyclase. The physiological response will be glycogen breakdown. Light activate rhodopsin, and its effector will be cyclic GMP phosphodiesterase for the visual excitation. And there are some more examples. You can see receptor showing 7 transmembrane helices. The G proteins are heterotrimers consisting of the alpha and beta and gamma subunit. It is the alpha subunit that contains GTPase activity. When G protein is activated by binding GTP, something like this, the heterotrimer dissociates into the alpha subunit and beta gamma heterodimer. Both alpha subunit and the beta gamma heterodimer can independently transmit the signal received from the receptor to different effector molecules. G proteins bind guanine nucleotide, hence the term G protein, and act as a molecular switch that are activated by binding GDP, are inactivated when the GDP is hydrolyzed to GDP. The hydrolysis of GDP is normally accelerated by regulatory molecule known as RGS. In human, more than 1,000 receptors are known, and also hundreds of different G proteins are known. So together, so many different combinations for different signal transductions are possible. You can see here G protein activated the adenylate cyclase by hormone binding. Hormone binding to the receptor lead to the activation of G protein. The replacement of GDP with GTP dissociate alpha subunit from beta gamma. GTP bound alpha subunit binds to adenylate cyclase and activate to make cyclic AMP. Cyclic AMP is a second messenger that affect a diverse range of cellular processes. Here we're going to look at the structural basis for signal transduction induced by G protein. Many interesting aspects shown here. Ras proteins function as a regulator of cell division and differentiation. Ras protein is kind of G protein, but it doesn't have any GTPase activity. Instead, it could be inactivated by binding to GAP, GTPase activating protein. Ras protein is activated in response to protein tyrosine kinase receptor. About 25% of all tumor cells have mutant Ras proteins that cannot be activated to hydrolyze GTP. As a result, these mutant Ras molecules remain bound to GTP and activated, and this leads to uncontrolled cell growth. The structure of Ras protein was determined by the Korean scholar in the United States, Sung-ho Kim in UC Berkeley, and Ken Holmes in Max Planck Institute. Ras protein is alpha/beta protein. Here you can see 6 beta strand and the alpha helices at both sides. You can see the topology diagram. The active regions are colored in red. Basically 5 loops, G1 to G5, together with the GDP bound. All small GTP binding proteins and G alpha subunit of trimeric G proteins have highly conserved amino acid sequences in 3 loops, G1, G3, and 4. You can see the conserved sequence for the G1, G, any, G, K, S, or T, called P loop or diphosphate binding loop. It functions as binding to alpha and beta phosphate. T3 has a DNE forming a link between subsites that binds to magnesium and gamma phosphate group. T4 has NK, NED, together with G5 serve as recognition and binding to the base. G2 function as the binding to the magnesium. Almost all nucleotide triphosphate hydrolyzing enzymes, including RAS and G-alpha, requires magnesium ions for their catalytic activities. Magnesium is presumably requires both for proper positioning of gamma phosphate for weakening the PO bond that is split during catalysis, as well as for maintaining the stability of guanine nucleotide complex. In RAS structure, magnesium has 6 coordination bonds with oxygens, 2 from GTP, 2 from water, and 2 from threonine and serine. Now it's time to figure out structural changes that convert resting G-alpha GTP to the active G-alpha GTP complex. 2 regions in RAS molecule undergo large conformational changes when the molecule switches from the active GTP bound form to the inactive GTP bound form. They are called switch regions. Are found in loop G2 called switch 1, and the region comprising loop 3 and helix alpha 2 called switch 2. Here we're going to look at the structural changes about the transducin, which is a kind of G protein. Light just activate rhodopsin in our eyes. The transducin is activated and leading to the activation of cyclic GMP phosphodiesterase. The receptor in this context is rhodopsin. Rhodopsin is very similar to bacterial rhodopsin we learned in the previous chapter, except that it can activate transducin instead of the protopumping in bacterial rhodopsin. Transducin has large 2 domains. The GTPase domain in red color is belonging to the RAS protein. The alpha helical domain in green color is kind of inserted big G2 loop in comparison to RAS protein. In this case, there are 3 switch regions colored in pink. Switch 1, 2, 3. GDP binding sites are positioned crevice between 2 domains here, buried region, so that the exchange of GDP and GTP is accompanied by the opening or closing of this region, induced by large conformational changes.